everyone. Hello, everyone. Welcome to today's seminar at Institute of Bioinformatics. I'm Chen Xie. It's my pleasure to introduce Dr. Sean Ryan. I met Sean at Plant and Animal Genomics conference last year when my advisor CJ took us there. And I was browsing the poster section and being bombarded by all kinds of scientific discoveries. But then I got attracted by Sean's poster about genome editing products regulation. That's how I knew Sean and learned about scientific consulting as a potential career path. But today, Sean's gonna tell us something also about scientific discovery. He got his PhD of biological sciences from University of Notre Dame. And he spent some time with USDA, NC State University, and University of Tennessee as postdocs. Now he's a managing scientist at Exponent and also an assistant professor at University of Tennessee. Sean's decade-plus career has armed him with insights into climate change impacts, invasive species, and the dynamics of ecological genetics. Today, he will share how citizen science is not just a buzzword, but the key to unlocking our understanding of species adaptability. So let's dive in, and please join me in welcoming John Ryan.
Just want to thank you, Jen, for inviting me, and I think the best host I've ever had in terms of coordinating and making sure I'm taken care of. So I really appreciate that, and just honored to be invited to share some of the work that I've done that I don't get to do so much anymore as a consultant. I'll tell a little bit about consulting at the end. If you weren't able to, we had a recruiting event yesterday, talked a little bit about Exponent consulting in general. I'll share a little bit about that at the end, but what I'll share today is the work that I had done prior to coming to consulting that I'm very passionate about, that I try to keep moving through collaborations and some hobby. But I think it's, well, obviously I thought it was important work that I was passionate about, and I think that it's hopefully relevant to you, and maybe in some ways you can keep the work that I had started going in your own way. So obviously I'll be talking about citizen science, some about genomics, evolutionary biology, and global change biology. I used to have a slide that would say, enter the Anthropocene, but apparently geologists have thrown a muck in that, and they don't believe we're in the Anthropocene. So, whether we're, truly in our own, geological period, doesn't escape the fact that humans have been dramatically altering Earth's ecosystems now for quite some time in a number of ways. So through our movement, through ourselves or products. So these are routes through air traffic and ocean shipping. We've altered the planet through changes in land use. It's probably the most impactful. Also alterations to disturbance regimes, such as like wildfire. I live in California. We're very aware of wildfire. And then obviously climate change. We're altering the Earth's climate. Sea level rise being one of those impacts of that. And so when I started my career, so a long time ago, PhD program in 2010, climate change was a hot topic, and I was very interested in how species were responding to disturbances such as climate change. And there was a good amount of research that was going into understanding what I would describe as the where species are, so how they're responding in terms of their distribution, and then in terms of their phrenology, so the timing of their biological activities, like when the flowers are blooming or birds are migrating, those events, we're getting a better handle on those responses. But in terms of evolutionary responses, we didn't really have a good idea of how species were responding to past changes or predicting how they might respond into the future. And that's partly because we just didn't have the data sets to really answer that question. And so for my PhD work, that's kind of what I was tasked with doing. So I'll just give a really quick overview here, because it's not the main focus of my talk today. But for my PhD work, I was working in a system with tiger swallowtail butterflies. So if you're not familiar with the eastern tiger swallowtail, it's probably flying around campus throughout spring and summer. It looks like this. It's indistinguishable. I didn't put the Canadian tiger swallowtail. So there's two butterflies. The western tiger distribution is, you can think of this yellow as pretty much the distribution of the western tiger. The Canadian tiger is where that distribution ends. further north is the Canadian. Canadian looks just like the Western. It's almost morphologically indistinguishable, but typically usually a little bit smaller. But there are a lot of other differences in terms of physiology, detoxification abilities, things like that. And so the question was, Mark Schreiber had been working at University of Wisconsin and Michigan State, and throughout his career, he had sampled this hybrid zone between the two species throughout Wisconsin and Michigan. And he had done that in the 80s. So there was a great opportunity for me to retrace his footsteps, collect along those similar transects, and then look at how the hybrid zone had changed over the 30-year period where I was now reevaluating that hybrid zone. So collected a bunch of samples, spent a lot of time in my car driving through Wisconsin and Michigan, and did some population genomics. This was rad sequencing, very hot, method at the time. So you're sequencing a randomly subset of the genome, which is allowing you to sequence more individuals for population genomics. That's what you want. It's more individuals, so you sacrifice, an extensive amount of the genome. If that's not necessary, you just do a subset. And so with that, what I did, I did some simulation modeling. So the two species distributions were largely determined by climate, so the length of the growing season. And so I did some simulation to look at how that distribution might change under future climate scenarios, and in particular, what had actually happened from the 1980s to 2010s, where I had the sampling periods, look at those predictions, and then look at the genomic data and see how it's changed. We showed that the hybrid zone was moving along with this increased warming, as we would expect, and that the hybrid zone was staying intact. It wasn't breaking down. The two species were kind of blending together, but those reproductive barriers were staying intact as they were moving. Interesting research, but I think what was more important than the research itself, at least for me as an insight, was that this was really cool to be able to go back into the past, dig into these museum collections that he had created, and then look at evolutionary change within our lifetime. But those data sets, like Mark Schreiber, doesn't exist in many systems, and having these data sets, it was a unique thing. I was lucky to have that, and even though I was lucky, I still was like, oh my God, your sampling isn't so great. Well, that's because he didn't know that I was going to come along 30 years later. So it wasn't really sampled with that intention, but luckily it was good enough that we could come up with a nice paper. But so what I realized was that we really need to think about how we're collecting from populations so that we can ask these questions. So the question is, what do we need? So if you know the song, sing along. We need long-term population level sampling. That's what I'd argue we would need if we really want to evaluate evolutionary responses to environmental change. So when do we need it? Well, we needed it, I would have said when I started these talks 30 years, now 40, 50, we needed it yesterday, really. So the problem is, I've been out of the game a few years in consulting, but I don't think much has changed. I don't think NSF has really probably funded the level of projects that we would need to really start gathering these data. So it's still, I think, a deficiency that we haven't been addressing. And if my work wasn't, exciting enough to say this is why we need to develop these collections, you can just take a look at Peter and Rosemary Grant's work. If you're not familiar with that, take a look. It's kind of fundamental to any evolutionary biologist training to learn about the work that they've done. Peter and Rosemary Grant, husband and wife, professors, adorable. But also, in addition to that, they've been working in the Galapagos Islands, dating back to the 1970s, collecting data on various species, but most famous for Darwin's finches. So Darwin thought that evolution happened over a very long period of time, centuries, millennia. What they were able to show through meticulous documentation of various phenotypes, in this case, beak depth, that they can actually witness evolution in real time within their lives. And so they were showing that beak depth was changing in response to changes in climate that was being mediated through changes in their food resource, and that had further implications for the ecosystems they lived in. So really incredible work. How do we take what they've done and scale it up, right? So instead of just doing Galapagos Island Darwin's finches. How do we get this so that we're doing more populations, more species at a greater scale? How do we do that so it's cost effective? Because, paying for them to go out into the Galapagos was probably pretty expensive. And that's one of the issues with long-term sampling is it's costly. So how do we do this at scale and not let them have all the fun either? And bring more people into this. So I would say, well, humans are part of the problem for, the Anthropocene that I think we're in, but they're also part of the solution. So it turns out there are hundreds of millions of people at the very least across the world that are interested in science, are interested in making a difference, are interested in the health of our ecosystems. Many of them have access to the internet or smartphones. And so this is a large population that we can engage in the scientific process. So bring them into the fold. And we can do that through citizen science. So citizen science, so how many people have heard the term citizen science or community science, some variation? So this is better than 10 years ago. It probably would have been like 2 people. How many people have been involved in a citizen science project or led one? A few here, that's pretty good, still even better. Yeah, definitely than 10 years ago. And so I guess I should say, what is citizen science? Maybe that'll change whether your hand goes up. The way that I've described it in papers and kind of how I think about it is scientific research that involves non-professional scientists. It's just a way of distinguishing between, obviously, if I work in a lab, my grad student's not a citizen scientist because they're involved in that research, they're getting a PhD or they have a PhD in that work. So it's just, inviting essentially people in society that are outside of your area of expertise into helping with your research endeavor. So I think if it doesn't really matter what we call it or how we define it, it's just thinking about collaboration at a more inclusive, broader scale, so bringing more people into it. And I think the best way to describe citizen science is Jennifer Shirk, who's been a practitioner in citizen science through Cornell's bird or ornithology department. Citizen science offers the power of science to everyone, the power of everyone to science. So there's not just an opportunity to engage the public to help collect data, people that can improve the science that we're doing. There's also an opportunity to educate the public. And when it comes to evolutionary biology I was talking about, there's huge opportunity there. It could be for vaccines and a number of things. So there's a benefits on both sides of this if the project's done well. And so citizen science has been growing rapidly. It's been around. So the Cornell Lab of Ornithology is, dates back like 100 years starting this. Did their work there. But most of citizen science was, that flavor that ecology would have been mostly with birds, but has expanded, and then astronomy. But since in the last decade, it's kind of exploded in terms of work in the environmental justice area, now human health, and even agriculture, genomics, evolutionary biology, and other fields as well. So it's kind of growing into multiple fields. Many projects, like examples here. Who's familiar with iNaturalist? Anyone here? It's just a great way to document what you're seeing, provide that data. But like the bucket brigade, people going out collecting air samples. There's Purple Air. I don't know if you're familiar with, it's monitoring air quality data. Projects to look at microplastics that are washing up on beaches. Leads to lawsuits, and I think this is like a $50 million, $500 million lawsuit. And so just really a nice diversity of projects. But I included agriculture, genomics, and evolutionary biology in here, but I'll tell you, as you'll see, they're still underrepresented. And so I'm just going to tell you a little bit about the work that I've done, just briefly about citizen science and agriculture. We'll come back to evolutionary biology and genomics in a second. And I'm going to share it just because this was just a great opportunity. And I think there's some insights that we gained from this work that I think are hopefully be valuable to you to share and are relevant to the broader context in which I'm talking about citizen science. But I was a postdoc. So after I worked at the USDA, or while I was there, I got a NIFA postdoctoral fellowship. It's like an NSF fellowship. It allowed me to do the research that I wanted, which you'll hear about in a little bit. I've got a little white butterfly, and go where I wanted. At the time, I wanted to go to NC State. I wanted to work in Rob Dunn's lab, who had done an enormous amount of work in the citizen science space out here. And while there, I led this team of just an amazing and diverse group of researchers, teachers, people in government, academia, industry, to look at the role that citizen science was playing in food and agriculture. And I won't name them all, but their faces are here. And so in that process, this is a paper we published back in 2018, was it? Yeah, 18. And it's kind of become a foundation in the field. I think it's a great read. I know I'm biased here, but I think if you're interested in this topic at all, it's a very fun read, and it kind of covers everything. But I'll just tell you a few of the results from that paper that I think are relevant here. So one, as I said, agriculture is underrepresented in the field of citizen science. And so if you look at SciStarter, which is a hub, so if you want to look for what citizen science projects are available, you can go on SciStarter. There are about 1,400 projects now. If you look at the tags they have for agriculture for food, you get less than 1% of the projects actually have those tags. And if you look at those projects, they're not really agriculture and food even, they just have those tags. So it's actually far less. If you go on Web of Science, let's look at published studies. 9,000 roughly papers use the term citizen science. Of those, 5% contain the term agriculture. If you dig into those, I'm going to bet that a lot of them aren't actually involved in agro-ecosystems in any way. That word just happens to be in the abstract. And so it's like, what's going on here? Agriculture is such a big area. Why do we have so few citizen science projects? Part of it is just because agriculture has been doing citizen science-like projects for a long time. They just didn't call it citizen science. This is just a new term. So that's part of the issue here. So in fact, some of the early citizen science projects ever were agriculture, like dating back to 3,500 years ago in China, where they're asking people to go out and make reports of locusts so they can use that information for kind of a pest detection and management approach. So there's a long history of citizen science in agriculture. It just wasn't described as that. The other problem is that the new projects in the field of citizen science, like I said, are focused mostly on ecology, astronomy, and so they're just not really thinking about problems in agriculture. And then the other problem is attribution. Sometimes the projects will involve the public or be doing citizen science, and they just don't give credit to the people that are contributing. So just a lack of attribution. But with that said, even though the term isn't used, maybe there's not the attribution, there's a wealth of examples of projects, citizen science projects, that are addressing some of the fundamental and grand challenges in food and agriculture. So monitoring for diseases. So actually, I'll bin these into the kind of think of these like the methodological approaches for citizen science projects. We have observational, collections-based, and manipulative. So the way that I describe them, so observational would be like a naturalist, you're taking a picture of something, or you're reporting something. Collections-based, someone's actually going out, collecting some material of some sort, like biological, and then maybe sending that in. This is one I'm very fond of because I think it's necessary if you want to answer many of the questions never looked at ecology that I would like to answer. But also manipulative, which I think are very powerful experiments. But the difficulty level kind of increases as you go down this list here. And so manipulative experiments, there's examples of studies in Finland where they engage 4-H students. So this is youth in agriculture. And they actually measured an ecosystem service, so the decomposition rate using like dung patties in field sites all across the entire country. So they did this at the scale that's kind of unimaginable, which is just an amazing paper in ecology. And then there's other studies in manipulative where we call tricot experiments, where farmers are given seeds of different varieties and then they plant those and they use, so it's the climate that they live in, the soil types, the management practices, and they can get a sense of how do those crops perform under those conditions, collect those data and send those back. And when I was recently at the Plant Animal Genome Conference, there's some really interesting work being done in this space since since we wrote this paper in Europe, where they're taking seeds from the seed banks that they have, all these different, varieties, and they're sending them out to people throughout Europe. They're going to expand maybe to the US, and then having these people grow them in their backyard and take measurements and send that information. So this, like, you think of as large scale phenotyping of these different genetic lineages that they just don't have information for. So just really cool stuff that's going on in this space. And then one other thing we talked about in the paper is technology. So the technologies, like the advances in technology have moved forward citizen science, just like they've moved forward science more generally. But it's some of the interesting ones that are relevant to agriculture, also to evolutionary biology and genomics is, the ability, gene editing get easier and easier and cheaper. So you have biohacking communities that are biohacking E. coli to produce vegan cheeses, trying to make biofuels and things like that. This is going on just in, cities across the world. Where this will go, I don't know, especially with regulation. Can you just genetically engineer something and then put it in your backyard? So it'll be interesting to watch how this unfolds and how this gets incorporated into projects like citizen science, where people are kind of doing their own thing. As well as AI. So AI has been kind of a game changer in some ways, because if you use iNaturalist now, you can see you can take a picture of something. You don't know what it is. Used to, you'd have to farm that out to other people. Maybe they can make a guess. Now AI will guess what it is based on your location and the image processing. So just it's an amazing in terms of taking what is data and transferring that into, or converting that into information in real time. Things like drones, and then even DIY, like 3D printing, is getting just easier and easier to collect data of all sorts through various sensors. And so just huge opportunities here to collect just a wealth of data at a huge scale when you have, let's say a fleet of citizen scientists that are going out and taking measurements as part of a particular project. And then one thing we wanted to highlight too was that the technology doesn't have to be sophisticated or expensive to be impactful. In fact, some of the best technologies may just be ingenuity, just to come up with something interesting, like having people wear a sock over the boot, walk through a trail, take that, put it in a bag, send it in, and then look at little pathogens that have been attached to their sock or something like that. So that's the best in terms of, really getting your project out at scale, especially in communities that might not have the resources to go and buy all this fancy equipment. And so let's travel back to evolutionary biology really quick. So I went and I did the same thing. I looked at SciStarter and said, well, what do we have in terms of genomics evolution? Well, first, they don't even have tags for these terms. So maybe we should send them an email. But they have just a handful of projects doing genomics related research, evolution related, so very underrepresented. That's also reflected in the literature. Very few of these projects seem to be doing work in genomics and evolution. Those that are in genomics, sometimes I wouldn't call them citizen science. Like, 23andMe is not citizen science. It's a paid for service, in my opinion. But there are a few in there where it's usually through gamification. So people are developing games where people might do things like that help with comparative genomics or things like that. So they're solving little puzzles, just like was done for Folded, if you're not familiar with that. It's protein folding. They used to farm that out to citizen science through games. Now they just use AI and machine learning to do that, because they've trained them based on all the people that have contributed. So with all of that said, there's citizen science, or evolutionary biology and genomics is kind of still underrepresented in citizen science. But like I said, there's huge potential, and there's huge potential to do the types of projects like I talked about to understand how species are responding to environmental change. And so when I started, there was this cool project looking at having people photograph snails to look at snail morphology and evolution in urban environments. That kind of gave me inspiration for the project I did. And then there was the Cornell Bird or Lab of Ornithology, work with a number of bird species, documenting their distribution and migration. And so here, theirs are just the most incredible. They get like 1 million observations a month or something. And then there's eBird, and this is eBird. And so they can do these incredible things like look at the abundance change across South and North America over time. And I looked at this and I said, well, I want to look at this and see evolutionary change over time. Can we see allele frequencies changing through space, across space and time? And the only way to do that, I think, is to do, probably engage the public, do citizen science, so we can do these things at scale, cost effectively. So that brought me to the project that I'm going to tell you about today. That's probably the project that I'm most fond of that I started as actually a side project. So this wasn't part of my dissertation, but it was something that I had learned like, God, this is something I want to do before I graduate. And I just went ahead and did it. So maybe I'm lucky in that it ended up working out, but I would say if there's something you're really passionate about, obviously your advisor will have some input on this, but don't be afraid to maybe, try to somehow weave that into what you're doing. And even if it doesn't get finished during your PhD, it's something maybe you can carry on into your postdoc. So just to recap, it's like I want to kind of know how species are responding to environmental change. And I want to create something long-term that we can look at, evolution over time. And so how do we get there? Well, we need, like I said, long-term population sampling. We're going to need a species that people can actually go out and collect. We're going to need a species that is relatively abundant. Obviously, this is not going to work well for an endangered species. And so what I came up with, as you saw in the previous slide, is the small cabbage white butterfly, or Pyrus rapi. Butterfly is found through many, many parts of the world, everywhere pretty much except for South America and Antarctica. And what's interesting is that we actually don't know we knew that it came from Eurasia originally, but it was introduced to North America in the 1800s and then early 1900s into Australia and New Zealand. So but we don't actually know what actually happened. It's an agricultural pest, so it feeds on plants in the mustard family or brassicas, and that's part of why it spread across the world. And so I thought this was the perfect system. The way that I would pitch this is you can't start a project like this with the goal of, looking at evolutionary response. If that's your first project, you're in trouble, right? Because you're looking at like 10, 20 years to actually get there. So the goal of this was to pick a system where I can answer an interesting question now, like within that short 2 to 3 year period, that is interesting and important, but at the same time, I'm building these collections that could be used for a long-term study. So the goal was to look at the invasion routes using the genetics or the genomic analysis of the butterflies that we were going to collect for this project. And so first step, build a web page, put all the information on how, about the project, how people can participate. We had some educational materials and fun things. It changed quite a bit. This is a process that's very iterative. It's kind of like a startup where you just like try things, they work, don't work, and you keep going. And then over time, just reaching out and bringing in more grad students and postdocs into the project as it grew to get involved in various aspects of the work. Then getting the word out to the public. Hey, we would like your help to help, collect this butterfly, send it in. So social media, it's back when I was on Twitter and Facebook. And then all these other, like we even were on Discover Magazine. They had an article about us and did interviews. All of this was great. We got a lot of attention, but what's interesting is that it didn't translate into sending in butterflies. Really the low-hanging fruit or the most efficient way to get people to contribute was to reach out to things like the like Ecological Society, or even better, entomological societies, because these are people that are already going out, they're familiar with butterflies, they may already be collecting them. So that for us was the low-hanging fruit. So just to mention that if you come up with a project, just getting the word out to everyone isn't necessarily going to translate into, getting people to participate or contribute. You really want to maybe think about who are the people that are most likely to contribute. Make sure you focus most of your effort there. And so with that, it was pretty successful. So before getting into what we were able to achieve, I had put together, we got quite a few samples from North America, through parts of Europe, and really good sampling in Australia and New Zealand. But we were missing these samples in Asia and some parts of Europe and even Hawaii. And I found out that Roger Villa, he's a professor in Spain, had postdocs. His postdocs had gone out and sampled, just happenstance, it was amazing, all the places we hadn't sampled within a similar time period for a different project he was doing. And so I found out about his work. we joined forces, he sent me those samples, and we combined them together to create, which is almost the entire species distribution for the species, which is incredible. So that's one thing I would say is that when I think about citizen science, I'm not a purist that like everything has to come from citizen scientists. No, you do, what is needed to move the project forward. In this case, it's a collaboration with the public. It's also a collaboration with this other researcher who had specimens as well. And so, the one fun thing is, a common question by people is, what didn't people send in the wrong butterflies? That happened. And guess who sent in the wrong butterflies?
Scientists.
Yes, people with PhDs. So if you get a PhD, you're more likely to send in the wrong butterfly. I don't know what's going on there. But I think, if some species in some areas, there can be, similar looking species, you need to know that and you need to make sure that people know what they're looking for. So that's just part of designing a good citizen science project is knowing where people might go wrong. But this is true of undergrads, postdocs, even, professors. You just have to think these things through and then make sure your methodology is tight. And so at the time, not only was I doing this as a side project, I didn't have any money. I had no funds. This was all just like, it costs nothing to build a website and ask people to send you stuff. And so it was a little bold, stupid maybe had it not worked out. Since it worked out, I can call it bold. And it was like, well, I'm just going to get all the samples and we'll figure it out, right? Someone's going to pay for this. And so back then, crowdfunding was really popular. Not so much now because I think people realize this is just a great way to ask your family for money. Or not a great way. And so that's kind of how it happened. But we were able, that's probably a good thing of having a bigger group, right? Is that we had more family members to ask. We did get some people, just like random people to contribute. But I think if we run the numbers, it's mostly friends and family. But we got about $6,000. And then with that, the beauty of that was that I was able to leverage that through our John Riley Center at Notre Dame and then the environmental change initiative that we had to get them to match these funds. So now we're really putting some money together. We could do an initial round of sequencing, get some preliminary results, apply for a NIFA fellowship, and then get some real funding. And so that's kind of the approach that I took. And then with that, we did 15 lanes of high-seq. So quite a bit of sampling, sequencing for about it was actually over 600. 600 made it into the analysis. And that allowed us to sequence about 22,000 autosomal SNPs. We also had sex-linked markers, but they weren't included in this analysis. And we also sequenced CO1 so the mitochondrial species diagnostic marker for about 1,000 of them. We have mitochondrial data as well. Some of the analysis in the paper. I'm just going to talk about the RADseq data. But with it, we were able to show that there are at least 7 genetically distinct populations throughout the world, and likely more. So if you go to Asia, so the way structure works, if you're not familiar, so each of these bars is an individual, and the percentage of their ancestry assigned to a particular genetic cluster, which largely corresponds with geographic location. And the thing though is it's saying that the best fit is likely 7 populations, but this is an art somewhat of as it is a science, and so you can push it to say, like, let me look closer at Asia, and here you see that this actually breaks down to each country is pretty much a genetically distinct population as well. So there's just not as much differentiation, but there's some pretty distinct differentiation in Asia. What's interesting is that I thought we could see that in Europe as well, but we don't. It's like one pan-mictic population for the most part. Why that is, why there's so much mixing going on, it's not clear. One thought is that there's some migratory-like behavior that exists, and so it may be that they're actually like, this semi or pseudo migration that's going on. It could be that there's just so much interconnection going on that they're just being shipped around and moved around. It's not really clear, but that was an interesting outcome. The other really interesting outcome is that in the US, there's this, we call it a pink population, but this Western population that's genetically distinct from everything else in North America that we weren't expecting to see. And it's like, well, what's going on there? So we'll come back to that. Before we get there, what we need to know is what we're trying to figure out is the invasion history. So you can think of I describe this as 23andMe, but with a butterfly. So we're looking at the ancestry, and then we're trying to understand how these populations diverge through time. And so here you have, this is what we're seeing today, and then we go back in time, we can look at how they branched. So the main hypothesis is that these butterflies started off somewhere in Eurasia. So there had to have been this branching between Europe and Asia historically. And then the question is, well, we now have this genetically distinct population in Siberia, essentially. Did it come from Europe or did it come from Asia? And so we can present those here in these trees here. So this would be Europe giving rise to Siberia, or is it Asia giving rise to the Siberian population? And so this is how you do what's called an approximate Bayesian computation analysis, where you have these scenarios, what you think may have happened, you can parameterize it with real data, which if we know the estimate of the data first arrival, whatever information we have is more informative, but you do provide what you have in terms of information. And then you model this. So you run these massive simulations. So what that would look like, what those populations would look like genetically if that were the case. And then essentially we compare that to what they actually look like. And so we can see what is the most likely scenario given our data. And so we did that. And the first thing we see is that about 20,000 years ago, huge confidence intervals, I'll just make that clear, right? Is that by 10,000 years. So we're not really exactly sure, but it looks like maybe 20,000 years ago, there was a large population expansion from this Eurasian population, which corresponds with the last glacial maximum. So this has been shown for a lot of other species in Europe. Ice was receding, and a lot of populations were exploding or expanding in response to that. And so that may be what's happening. It could be that maybe the lower end is more accurate, and that corresponds with more cultivation of its host plant or something. But this is our best guess at the time. And then about 1,200 years ago, we estimate, or the most likely scenario here is that out of Eastern Europe here, or the Levant region, we see that the European and the Asian population diverged from one another, and this corresponds to the peak in trade along the Silk Trade route. So there's an increase in trade during that time period, and also corresponding increase in the morphological diversification of brassica crops. They were getting leafier varieties. So maybe the combination of those two things were helping with the spread of or divergence of the species into Asia. So nonetheless, very interesting. And then in terms of the Siberian population, it actually corresponds really well to the development of what were called the Siberian trade routes. So through Asia during this time period, about 1700, there was formal trade routes that were established between Asia and Siberia, and that's likely what allowed these populations to be introduced to the region. This population, just want to mention, is really cool because it's in Yakutsk, which is one of the coldest cities on the planet. So clearly these things have no problem with that. And then North Africa. So I couldn't find any actual first observations. So what we usually try to do when we're reconstructing invasion biology is like, when's the first time someone saw this? Well, I couldn't find any accounts in North Africa, but we estimated that it was about 200 years ago. And that would correspond with French colonialism in North Africa. So perhaps through that process, the cabbage white was introduced to North Africa and has since been kind of diversified into a distinct population. Although it is possible that this date is even more recent, but we're not sure. And then in terms of North America, so just as we expected, these populations did come from Europe, but we weren't sure whether there was a single introduction or multiple introductions, like waves of introduction. And then in terms of our approximate Bayesian computation, comparison of different scenarios, it's kind of, it's a coin flip, but based on other information like the mitochondrial DNA that we sequenced and other things, it's pretty clear that there were multiple introductions. There had to have been at least 50, probably many more than 50 individuals And I'm thinking on the boats that were coming across, that's probably, you needed at least a couple of trips to get there. We also see quite a bit of genetic diversity in the North American population. So with each introduction, usually you get a bottleneck, right? You go from this large population to this founder population, really small, you lose a bunch of genetic diversity. We see in North America is that North America still has quite a bit of genetic diversity, which multiple introductions would probably favor that. And there was also an entomologist, Samuel Scudder, who had retraced. So he was around during this initial introduction, and he actually performed a citizen science experiment. Obviously, they didn't call it that back then. He asked people from all over and said, hey, there's this new white butterfly that we've heard of. Can you send me what you've seen, like either your observations or the actual specimen? And he actually reconstructed that there was probably multiple invasions, and then he documented it spread across North America. Now, what we saw was that for this pink population, we're predicting that it happened pretty recent after this initial introduction. So that I said, I didn't say here, but it was the 1860s. That was actually like Darwin writing The Origin of Species. Civil War is about to happen, and shortly after that introduction, we predict that this population formed. So Arthur Shapiro, who studies butterflies in California, has done a lot of work. Well, maybe it's the Spaniards brought it earlier, but based on this date that we're estimating, that's like that doesn't seem to be the case. They weren't brought by Spaniards. So how did this genetically distinct population form and form so quickly? Any guesses? How they got there? Anyone know what was happening in the late 1800s across? What's that? Not the Oregon Trail, but there was some transportation that was developing that many of us don't use anymore.
Yes.
It's not for our personal. What's that?
Railroad.
Rail lines. So rail lines were being developed extensively during this time period, and I looked into, well, what did that look like? And I was fortunate to find some grad student that had been piecing this together. So he put together rail line development over time. So we have early 1800s. This is how it was developing. And what we see is something happens just before we predict the butterfly to be introduced to California. There's this connection from East Coast to the West Coast, specifically the same region that we're seeing this pink population. So my guess is it took a train ride to get there, which is really cool because I think that's as close to a smoking gun as you can get. It's like, I think this is what happened here. So a fun story for them. So they're not just taking boats, but now they're traveling by train. They're doing it all. And then from there, they were introduced to Hawaii. And then the next question was, well, how did they get to New Zealand and Australia? And the thought was like, well, maybe they came from England because, they were a former colony of England. And there's a lot of trade, but there was still about 10% trade with the US. And it turns out that we actually gave these butterflies to New Zealand, and like good neighbors, they then passed them off to Australia. And so interesting turn of events that it wasn't clear until we had done this analysis of who was responsible for each of these introductions. And so I just want to say a few more things about this project that I thought were interesting. Like, okay, so we have this population in the West Coast. How extensive is this? Like, how far does this go? We don't know. We've had more samples since then, so we could go back and do some more sequencing to look at this, because we have evidence of admixture or gene flow between them, but we don't have any like F1 hybrids in here, but they probably exist. And like, how are these things even different? Like, is there anything interesting, different about them? We don't know. So there's a lot of interesting things that could be done here. And then one other interesting thing that I didn't publish this. I just worked with some undergraduates and even a high school student. I should publish it. But what I did was I was doing this project and everyone said, well, what about like viruses or endosymbionts? Did you see that in your samples? And I said, well, I did RAS sequencing. It's not really ideal for that. So I don't think I would have picked it up. And it's like, well, you should check. So what I did is I took all the reads that mapped to the virus rape genome, because we had a genome at that time. Should have mentioned that. And so I just threw those reads out. That's not what we're interested in. But what remains? So they things like parasitoids, host plants, endosymbionts, pathogens. And so what do we find? So if we look at prokaryotes, we see some interesting things like Wolbachia and a number of individuals. So Wolbachia is a very common endosymbiont in insects. It's being used sterile technique with mosquitoes. Really interesting stuff going there. We can talk about if you want at lunch. And then BT, even finding BT on these butterflies or in these butterflies. And then a bunch of stuff also that's related to reagent contamination that's probably in every lab. So also interesting, we look at the plants, it's kind of a who's who of agricultural crops. Not surprising, these things live in agricultural ecosystem, but kind of cool that they're picking up some DNA of the plants that they're visiting. Again, because this wasn't intended for this, you can't really do a whole lot in terms of like, well, you know, visiting them more or whatnot. There's just too much noise and all of this. And we look at fungi. What we see, what I think is interesting of this, some of the species we're seeing is plant pathogens. So are they picking up plant pathogens? Are they spreading them to the plants that they're visiting? No idea, but it's interesting that we found some on them. And then the most interesting, which is like kind of blew everyone's mind, that it had me scratching my head. I was like, well, what animals? I was expecting to see maybe some parasitoids or something. Maybe there was a parasitoid in one of these things or somehow something interesting. What we see instead are things like we see carp, we see mouse, we see fruit fly or fruit flies, well, Ragalides. We see zebrafish, chimpanzee. And so what's going on here? Anyone have a guess? Was it? Lab contamination. So this is a who's who of what's being studied at Notre Dame at the time. So this is our genomics score wasn't happy about this. But I imagine if you look at any genomic data sets and you dig deep enough, you're gonna find this. And what's interesting, like I don't know if you can use it for anything, but if you ever want to do forensics and know where that DNA sample came from, you could probably use these as your diagnostic markers to say like, you know, what's the community look like? This is Notre Dame. These are all the species they study. UGA probably has its own unique set of individuals. Of course, there's going to be some overlap, some non-informative markers, so to speak. But it's just interesting that it's like, wow, we found all these interesting species from other labs. Just trace amounts of DNA making it into our samples. And then one last thing I'll say here is that not only do we do these genomic analyses, but I wanted to use everything we could because, you know, I do feel bad killing butterflies. And some of our social media pages, people would be like, you want me to tell my daughter to go out and kill butterflies? Yeah, but I didn't say it like that. It's agricultural pest. If you eat broccoli, you're paying someone to do that for you. But yeah, I understand. I get it. But so we wanted to kind of make the most of all these collections, right? Make their lives matter here. So let's can we get more information? So I was working with Anne Espesat, you saw earlier. She was interested in terra, this white pigment that's produced. That's actually supposed to be an honest signal in sexual selection. So the males are actually the ones that are pretty, as usual. And so they reflect more with this white pigment. So the females can see them, and it's the more nitrogen in their diet, so maybe the more healthy they are, the more they reflect, the more that pigment they produce. So she was looking at that and seeing if she could find some correlation with land use. I think the problem is that's just too noisy, but she was doing another number of other projects with that as well. But that was the hope there. And then I was working with Andrew Sower at Butler University in Indiana, and he was really interested in how these traits, like the amount of melanism and the size of the butterfly and this dark spot here, changes with photoperiod, like day length and also temperature. And so I was thinking, like, well, I created a citizen science project on Zooniverse where you can put photographs on there, and then they have all the tools where you can, you know, basically customize it so you can have people do whatever you want in terms of making measurements, and then they'll farm it out to their community. They'll take these measurements for you. You can pick how many people you want to do the measurement, 50, 100, whatever you need. So you can get really good estimates of whatever you're looking at, like size of the butterfly, and then we're going to use that information. I think it'd also be just interesting to look at how the phenotype of this butterfly varies across its distribution. And then just to say, like, I set this all up, it's kind of a bait and switch here, like, I want to study like how species are responding to environmental change. Let's do citizen science. But I haven't shown you any of that. And that's because it takes time. So now it's been about 10 years since I started this. The goal is I need to get out there and I need to cheerlead to be like, hey, everyone, let's go collect again. Because we have this great snapshot from 10 years ago. Let's do it again. And then in another 10 years, let's do it again. And so, it could be that, if you want to define, maybe an easier way to do this is to pick just a few locations and then have really good longitudinal sections for those. But the key here is just like, let's get out again and let's keep collecting this long term. And then the silver lining of all this is we have this natural experiment. We can also use these data sets to look at, without even doing any more sampling, we can look at genes under selection for adaptation. Maybe they're using different host plants. We know there's some evidence of maybe using garlic mustard. Maybe there's some evolutionary change there. I think, they're spread out all across this area. I think they're genes involved in diapause. So when they're going to go into dormancy, likely varies because photoperiod varies tremendously across this area. Maybe they're, in Siberia, there's probably some differences as well. So a lot of interesting things we can look at there. And then, I don't know if I really have time for that one, but just really quick that, just really interested in citizen science. And I wanted to see if I can get more people involved in collecting data. This would be more of a manipulative experiment where I worked with schools, got them to, just jump ahead here, I grew the plants for them, put in a temperature logger, worked with, this was first and third graders, but I also then worked with the Museum of Life Sciences in Durham, North Carolina. And people were actually willing to pay $20 to participate in this. So there's some potential funding here. At least pay, you're gonna get rich probably, but you can at least pay for your research. And they take them home. And then the idea is they record these different life stage events and the temperature each day. And so we can calculate what are called growing degree days. How much temperature need to accumulate for them to reach each of these life stages. And then with that, we can build really cool models. So here, this is going to be, I took that data, it's crude. Okay, bear with me, it's crude. But it's just like, we're going to take that estimate of how much temperature is needed before they trigger into the butterfly life stage. And what you're going to see here is not a migration. It's not a movement. Think of it as each pixel in this graph is like the wave. They're doing the wave. So they're saying, we're flying around and then they're going to disappear. And then they're going to come out again for a second generation because these insects have multiple generations. So we predict, if they live in those locations, they don't live in all these locations. But if they were to live there, this is what it would look like, is that they're coming out. And then you start to see, so that obviously in the south, they come out earlier in the year because it warms up quicker. And then in the north, you see that it's just starting. So they're already on their like third generation in the south, assuming that they're in those locations. They're actually not in Florida very much. But in the south, they are, like in this area. And so you can see these differences in the timing of when they're coming out and flying. And I just thought, this is so cool. We could engage the public, collect this data because we don't have it for most species. And we can do a better job of predicting the future, predicting when these butterflies are going to be flying around for multiple species or other insects as well. And we can do things like also look at what stage. We can ask people to cover these things and say, hey, can you determine what stage they become sensitive to the environment? And could you put different host plants in there and see if they perform better on one or the other? And so there's all this opportunity to engage the public and the type of work that I think they're really interested in. And I know I'm running way out of time here, but just a few things. So I work at Exponent now. So I did all of that. Now I work in consulting. And I was told I was the first consultant maybe here for this group. So you're like, what does a consultant do? Well, I can tell you more at lunch since I won't have time to get too much into it. But as a company, we work on all kinds of problems. So these are scientific problems. This is different from management consulting, where you might be helping companies with, management related issues. We solve their scientific problems, and or often involved in communicating the science to regulators, helping them get through regulatory approval. So that, what does that mean in terms of projects we do? It means that if it's for our company, because we're premier consulting, we work on difficult problems. I tell people that that means that if it's in the news, we're probably involved. So let's say a bridge collapse, maybe a plane window or a door flies off, things like rocket fails, we're probably involved in those things. But we also get involved in autonomous vehicles. We get involved in all types of construction related issues. And then from the environmental side, the things that I work on as an ecologist, I would put in terms of kind of evaluating the potential past and future environmental impacts. So on the reactive side, so litigation, someone's being sued for an oil spill, for the environmental ecological damages that it may have caused. My job is to try to figure out what actually were those impacts. Or let's say a wildfire comes through California that's not naturally occurring, and it was maybe a utility company or something. The question, there's going to be lawsuits. The question is, well, what were the actual impacts? So get involved in trying to determine the science there. And then what we would call proactive work would be clients have products, the ones I'm most interested in, and trying to do more those related to genetically engineered organisms. So say you have a genetically engineered fish, like Aqua Bounty's Aqua Advantage salmon, or a genetically engineered pig. The genus is a company developing a disease-resistant pig. Those have to get through regulatory approval for animals, so they go through FDA. Or crops, they would be USDA likely, maybe EPA, depending on what you put in there. And so my job is to evaluate what is the potential risk. And we work with regulators to figure out what those risks are and, if needed, potentially mitigate those risks. And so really interesting, every problem is a unique problem that we're dealing with. And then I'll end with my favorite collaborator on all my citizen science projects now, which is my daughter wearing a tutu in a lab. I don't know if that's appropriate, but this is my wife's lab, so. And then obviously funding agencies.
We have time for 1 or 2 questions.
Yeah, so, most land grant universities are gonna have like 2 big services that they offer that I think of. Like the first one is like soil and water testing, and then the other big one is some kind of plant diagnostic clinic, and that one's gonna be looking at anything from like plant molecular, nutritional health, and pathogens, insect pests, that sort of thing. And so thinking about the kind of forms that they have to turn in when they submit a sample, like that's a huge reservoir of data and historical data. And like, I thought about it and I was like, is anyone looking at that at a meta scale? Like, is anyone using that metadata and integrating all that data? Cause like, that would be super powerful. Cause those services have been around forever.
Yeah, you're talking about like extension.
Yeah, yeah.
Yeah, so I should have mentioned that, although I already went over time without mentioning it, but one of the things in the paper that we say the low-hanging fruit in the food and agriculture is exactly what we say. So you're making a very good point, is that extension agents exist all across the US. They have a really good reputation with rural communities, which are themselves communities underrepresented in citizen science. And obviously agriculture is somewhat underrepresented. And so we have this infrastructure in place that we should be leveraging. It's also that extension is unfortunately losing money over time and have experienced its own problems. And so there may be a really good opportunity here to do exactly what you're saying. So I wish you luck as you do that, because I think that's exactly what's needed, is to leverage those resources that have been there forever, but could benefit from kind of tapping into it and revitalizing some of what's there.
Okay, so you don't know of any universities that have tried to do that, or any USDA labs, or?
No.
That's insane. I mean, all that data just sitting around, that's insane. Okay.
I mean, someone might not be aware,
Right,
but just leveraging that resource,
sure.
I think is definitely under tapped.
Okay.
If not untapped.
I think let's let
Let's let Anne ask her question.
Hello.
Hello.
We can hear you.
Okay, all right, good. Just quickly, is there any way that somebody who's not officially a part of one of these things can access the sampling that, oh, you guys didn't look at, you looked at the charismatic megafauna, but I'm kind of interested in the microbes, and being able to, are there reservoirs or archives of things in any of these contexts, or oceanic or terrestrial, where people are sharing the specimens themselves with people who are not necessarily parts, analysis for not necessarily part of that project.
It's gonna, just like all science, is gonna vary by who's running the project, to some degree. But like iNaturalist, you can get their data, that's just observational data. I didn't mention Cal eDNA was a program throughout California, multi-institution. It was like Davis, Berkeley, I think UCLA. I may get it wrong, all the universities involved, but they were going out and collecting eDNA samples across California. I think all that data is publicly available. My data is publicly available in the specimens. If you want them, I'm welcome to use them. Send me an email. I think that might be true for many projects. I'm actually collaborating with the gentleman that was helping with the ABC analysis, is using my samples to look at purge of genetic load during invasion. And so I think it's just a reach out to these programs, and they're likely to collaborate with you or share their data, most likely.
Yeah, I wasn't thinking about the data, I was thinking about the actual specimens.
The specimens too, yeah. I mean, I can only speak for my project. I'd be happy if someone wants to pick it up and move it forward. I have a freezer full of dead butterflies in my garage that I'd be happy to put in an institution somewhere, and they can really get more use out of them than I am.
Yeah, thank you. This is great. I really enjoyed it. Thank you very much.
People who signed up for the lunch, we will move to the other room.